DNA Similarity and Classify. The DNA Similarity and Classification is a REST API that utilizes InterSystems Vector Search technology. Its primary aim is to identify genetic similarities by analyzing a vector database derived from interpreting a dataset obtained during the implementation of the project. Human DNA Sequences Dataset installation. To download the project, use the following command in the terminal: get clone. To run the project, use the command docker compose up dbuild. Attention! The project execution process can take a long time due to the DNA gene vectorization time between 30 and 40 minutes. Solution: Vector search. The system is capable of receiving a DNA sequence as input and, through iris vector search, identifying DNAs with similar genes. This functionality enables rapid identification of patterns and similarities among the provided genetic sequences. Initially, a long biological sequence is partitioned into words of length k with overlap. For instance, when employing words of length 6, referred to as hexamers. Consequently, the example sequence is divided into 4 hexamer words. While hexamers are utilized here, the word length is arbitrary and can be adjusted to accommodate specific requirements. Both the word length and the degree of overlap must be determined empirically for each application. In genomics, these procedures are commonly known as k-mer counting, where the frequencies of each possible k-mer are tallied. In this project, k-mer counting serves as a foundational technique for processing genetic sequence data, facilitating the comparative analysis of DNA. DNA sequences are vectorized based on k-mers, utilizing iris vector search for storage and retrieval, enabling the identification of DNAs similar to the received DNA within the database. Machine learning. Furthermore, the system provides a solution for processing and analyzing genetic sequence data using machine learning. Definitions are provided for each of the 7 classes, along with their respective quantities in human training data. These classes represent functional groups of genetic sequences. The classifications for each class can be found within the README file in the project. This sequence diagram illustrates how a system processes DNA sequences provided by a user. The API sends the DNA sequence to 2 components: DC, data, Humana for similarity search, and DC, data, NA classification for classification using a machine learning model, ModelMol. After processing, the API returns similar DNA sequences along with their classifications and probabilities to the user. To visualize the practical functioning of the project, we utilize the API available at which requires a DNA parameter. Upon making the request, whether through a browser or applications like Postman or Insomnia, the first 5 DNA sequences containing any fragment corresponding to the 6 parts of the provided sample will be listed. Below these sequences, it's possible to view the likely classification of the sample and the probabilities associated with other classifications. By conducting the search, existing similarities are more visibly detected.